Hello, everyone. Thank you for standing by. Welcome to today's Labcyte webinar on a new approach to early ADME CYP450 inhibition profiling using the Echo Liquid Handler. If you are experiencing difficulties joining the webinar session, please dial 800-263-6317 or visit support.gotowebinar.com. During the presentation, all participants will be in listen-only mode. To ask a question at any time during the event, you can use the questions window located on the right-hand side of your screen. Simply type your question into the window and click the send button. It's now my pleasure to introduce Jing Wang, an application scientist with Labcyte. Jing has worked in the drug discovery field for 15 years. She was a member of an ADME DMPK group responsible for advancing 15 compounds from high-throughput screening hits to INDs. She has comprehensive knowledge of in vitro ADME assay design and development, as well as small molecule enzymatic assay characterization. Thank you for your participation, and thank you to Jing. And we're ready to turn it over to her to start her presentation.
Thanks, Joe. Thanks, Joe. Hello, everyone. Thank you so much for coming to our webinar. Today, I would like to introduce a new approach to early ADME profiling, particularly in CYP450 inhibition by using the Echo Liquid Handler. So what is the ADME? Why do we need to evaluate ADME property early? So for those of you who are not quite familiar with ADME scientists study how drugs are absorbed, metabolized, distributed, and eliminated. ADME data provides key insight into what the body does to the drug. Their properties predict PK behaviors and drug-drug interaction potentials. In the late 90s, 40% of total drug failure was caused by poor ADME properties. Since then, ADME evaluation has been moved upwards into early drug discovery research. Multiple assays with large number of compounds are being screened in parallel with cell-based assays. Only compounds with potent efficacy as well as favorable ADME properties are advanced into clinical development. So in the past 10 years, the overall quality of drug candidates have improved dramatically. Now less than 10% of total drug failure was contributed by poor ADME properties. Early evaluation is expected to not only improve overall drug quality, but also shorten the drug discovery and development process. When a drug fails early, it fails cheap. This is why it is extremely important to keep ADME assessment as early as possible. However, running ADME assays in early discovery stage still has a few hurdles to overcome. For one, liver enzymes and cells are DMSO sensitive. The assay volume has to be quite large to compensate compounding DMSO, or compounds have to be diluted in buffer first to accommodate smaller assay volume. Some ADME assays are still being run in 24 or 48 well plates. The cost of profiling ADME early with a large number of compounds can really add up to the expense of a drug discovery. This is why it has become one of the top priorities for many pharmaceutical companies to find balance between discovery of best drug possible and cost saving. Here, I just want to show you an example as how much it will cost on ADME assay in early drug discovery. For example, cytochrome P450 inhibition assay. Cytochrome P450 enzyme, also known as CYP450 enzyme, is a large and diverse group of enzymes that are involved in drug metabolism. There are many CYP450 isozymes. 7 of them are responsible for metabolizing majority of xenobiotics. So the inhibition of these enzymes could be the major source of adverse drug interaction. This is extremely important that CYP450 inhibition is evaluated as early as possible. So in CYP inhibition assay, if you have a 20 microliter reaction, you are expected to spend about $8 per compound per isoform. If you run 320 compounds per week or 7 isoforms, you can expect to spend over $18,000 per week. In fact, I know many ADME groups are running 100 to 300 microliter reaction for this assay. The cost for running CYP inhibition assay is very, very high. Another important CYP inhibition assay is a time-dependent inhibition assay. Compound with a time-dependent inhibition could cause clinically relevant drug-drug interaction. For running time-dependent inhibition in 100 microliter assay volume, you're expecting to spend $27 per compound just for enzyme alone. If you run 20 compounds per week, you can expect to spend $540 per week on human liver microsome. There are many other ADME assays to evaluate drugs absorption, distribution, metabolism, and elimination. The cost for running all of these assays with a large number of compounds will be astronomical. So one way to cut cost, we believe, is to reduce the assay volume. Today, I would like to introduce to you our new approach to early ADME evaluation, which is assay miniaturization. With the traditional liquid handling, dispensing less than 1 microliter is almost always unreliable. There is a huge risk of a cross-contamination and carryover from transfer reagent with tips, nozzles, and pin tools. And for different fluid classes, for example, buffer with glycerol, usually require different calibration on your instruments. Of course, there is a cost of the tips. For echo liquid handler, however, we can transfer as low as 2.5 nanoliter with a high accuracy, and there's no cross-contamination and carryover. Echo can transfer broad range of fluid without the need of user calibration. There's no tips involved. Echo use focused sound energy to transfer. Focused acoustic transfer is one of the feature on Echo. Echo use sound energy to transfer as low as 2.5 nanoliter fluid from source to destination. There's no pipette tips, pin tools, or nozzles involved, which provides unsurpassed precision and accuracy, where it eliminates contamination, carryover. Destination place is inverted, and surface tension holds the fluid inside well. Echo can transfer 200 to 500 drops per second. Multiple drops gives large assay volume. Another important and unique feature on Echo liquid handler is dynamic fluid analysis. Dynamic fluid analysis enable Echo to analyze the content of the well, measure the height and meniscus of each well immediately before each transfer. Source energy is constantly being adjusted to ensure the best and most accurate transfer is executed. And with the dynamic fluid analysis, there's no need for user to calibrate, and Echo can transfer a wide range of fluid without the need of a user calibration. For example, Echo can transfer a compound in DMSO, reagent containing up to 50% glycerol, buffers, and cell culture media. Enzyme, for example, in ADME area, you can transfer human liver microsome, recombinant enzyme, or substrate. Echo can transfer all these reagents from any well to any well easy with the help of our software application. So Labcyte offers 3 easy-to-use software application. We have a play rate format, which can be used for replication and reformatting, transfer reagent from any well to any well. Echo cherry pick can be used for hit confirmation from multiple sources. Echo dose response software can generate IC50 or EC50. Our dose response software allows the user to generate IC50 or EC50. And IC50 can be generated in multiple directions, so energetic interaction can be evaluated. And a dose response software can also be used for direct dilution to eliminate mistakes and carryover from a serial dilution. Our software helps simplify Echo's application by guiding the user through key decisions. So with Echo nanoliter transfer, with Echo's nanoliter dispensing capability, dynamic flow analysis, and Echo software application, last year we have successfully developed miniaturized 1 microliter CYP450 inhibition assay. In this assay, both human liver microsome and recombinant CYP can be used as your enzyme source. We use luminescent substrate for its ultra-high throughput purpose. Of course, I recommend to pick most specific substrate for CYP of interest when you use human liver microsome and luminescent substrate. For assay development, we choose to test CYP3/4 activity with the luciferin IPA as a substrate for proof of concept purpose. So CYP3/4 is probably the most important CYP enzyme in the drug metabolism. It consists of greater than 30% of the total CYP in human body. The enzyme is involved in metabolism of approximately half of the drugs that are used in human today. So here is a procedure that most people use to run their CYP inhibition assay. The compound is first diluted in DMSO to create 10-point dilution. This DMSO stock is further diluted in aqueous buffer. This procedure sometimes has to be repeated twice to further decrease the DMSO concentration. Then 5 microliters of aliquot is taken out from this DMSO aqueous mixture, added to 5 microliters of the enzyme. Enzyme and compound is pre-incubated, and at the end of the pre-incubation, 10 microliter substrate is added. This CYP reaction is further incubated for appropriate amount of time. At the end of the incubation, luciferin detection reagents added and plays are rated on the reader. In the miniaturized CYP inhibition assay, we make 10-point compound dilution on echo. We use direct dilution rather than serial dilution to minimize the carryover or sticking to the pipette tips. We take 7-point nanoliter aliquots from compound dilution. With echo, here you're saving 600-fold reduction in compound. We add compound to 500 nanoliter of enzyme. With echo again, here you have a 10-fold reduction in enzyme. This mixture is incubated for appropriate amount of time. Then 500 nanoliter substrate is added to enzyme and enzyme and inhibitor. Here you have 10-fold reduction in substrate again. After enzyme, substrate, and inhibitor are incubated, the mixture is added to luciferin detection reagents added to enzyme substrate inhibitor mixture, and the plate is rated on the reader again. So with the miniaturized P450 inhibition assay, all of these procedures can be done on echo liquid handler. So this slide shows the data comparison between conventional assay format and miniaturized assay. Plot on the left shows IC50 determination on 4 compounds against CYP3A4 in the miniaturized assay. Each point on the curve is average of 8 replicates. As you can see, miniaturized assay generates superb quality data and excellent reproducibility. The table on the right simply shows the comparison between IC50 generated in our miniaturized assay with the literature values. These 4 compounds have been well characterized by many. As you can see clearly, our value falls within the bracket of published values. So we know CYP inhibition assay works in a miniaturized assay form. Next, we wanted to test our assay miniaturization with a larger set of compound and multiple isoforms. Earlier this year, we collaborated with Merck Serono. In this collaboration, we evaluated 7 different recombinant CYP450 isoforms and generated 10-point IC50 curves on 32 compounds. The IC50 generated in the miniaturized assay format were compared with IC50 value from 20 microliter reaction volumes. So this slide shows one of the examples from the collaboration. First, we were able to achieve consistent signal to background in the miniaturized assay when compared with a 20 microliter reaction volume. On this plot, x-axis represents IC50 generated with a 20 microliter reaction volume. The y-axis represents IC50 generated in the miniaturized assay with the echo liquid handler. As you can see, most compounds show similar IC50 with threefold variation between the 2 assay systems. A few compounds show more potent inhibition with echo liquid handler. The reason for that, we think, could be 1, acoustic transfer versus pipette tips. Some hydrophobic compound could absorb to tip surface and lost during dilution. This could for sure cause inaccurate IC50 with a pipette-based liquid handling system. Also, precipitation during dilution. When compound diluted in aqueous buffer first, then transferred into the assay, compound could be precipitated into the aqueous buffer. Therefore, it causes unreliable IC50. So similar findings have been reported by a few other pharmaceutical companies. In BMS case, when close to 1,000 compounds were screened, 10% compounds show potency only when echo was used as a liquid handler, where in AstraZeneca case, some compounds show greater than 200-fold more potent when echo was used as their liquid handling system. So I find these kind of data most compelling because without the echo liquid handling, not only there's a chance that best compound could be rejected during selection because their physical chemical properties, but in some case, time and millions of dollars could have spent on modifying the entire SAR around inadequate scaffolds. So early, I've shown how much it would cost for running CYP inhibition assay. It will cost over $18,000 per week to run 320 compounds per week. In the miniaturized assay format, you can expect to spend 42 cents per compound per isoform. If the same number of compounds are screened against 7 isoforms, the cost is less than $900 per week. With echo liquid handler, you can save over $17,000 per week for CYP inhibition assay. Next, I will talk about time-dependent CYP inhibition assay using mass spec as our detection method. Again, for this test, we use CYP3A4 and CYP1A2 as a proof of concept to study enzyme activity. So time-dependent inhibition refers to change in potency during in vitro incubation or in vivo dosing. The most common assay to assess time-dependent inhibition is IC50 shift assay, in which compound potency is generated and compared with and without enzyme cofactor NADPH present. When compounds demonstrate greater than 5-fold more potent inhibition, there's a chance the compound has a time-dependent inhibition potential. So goal to determine time-dependent inhibition is to identify compounds with mechanism-based inhibition. Mechanism-based inhibition is a severe case of a time-dependent inhibition in which compound could irreversibly inactivate CYP enzyme and cause idiosyncratic effect to human. Mechanism-based inhibition assay is one of the FDA recommended assay. The common assay to assess mechanism-based inhibition is a KI and K-enact assay. So for time-dependent inhibition assay, we choose to use rapid-fire mass spec as our detection method for its ultra-high throughput purpose. For this project, we collaborate with Compara Discovery Research Organization, a fantastic CRO company here in the Bay Area, specializing in bioanalysis. They helped and provide us with a high-quality mass spec data. So here's a procedure for running time-dependent inhibition assay. On the left, we have a procedure that are currently used in many ADME groups. Again, compound diluted in DMSO. Then 1 microliter aliquot is taken out from a DMSO stock and added to 100 microliter human liver microsome at 10x. Enzyme and compound is pre-incubated for 30 minutes. At the end of the 30 minutes, 10 microliter aliquot is taken out, added to 90 microliters of substrate. Enzyme and substrate and the compound is incubated again for 15 minutes. At the end of the incubation, the reaction is quenched with acetonitrile and plate is loaded onto mass spec. So recently, we have developed miniaturized and much simplified procedure to run time-dependent inhibition by using the echo liquid handler. Here, again, we use echo to do direct dilution for to create compound dilution and 10 nanoliter addition. So 10 nanoliter aliquot was taken for the compound from a compound stock, added to 2 microliter enzyme. For compound, you're saving 100-fold reduction. For enzyme, there's a 20-fold reduction in using miniaturized assay format. Enzyme and compound is pre-incubated for 30 minutes. For 30 minutes. At the end of the 30 minutes, 20 microliter substrate is added to enzyme and compound. For substrate, you have 20 microliter, 20-fold reduction in usage. Again, right after enzyme substrate is incubated, the reaction is quenched and then loaded onto mass spec. So for the miniaturized time-dependent inhibition, enzyme compound dilution and compound addition and human liver microsome addition is added with echo liquid handler. So here I show the echo workflow for running time-dependent inhibition with the help of echo software application. Here is your source plate containing test compound. You can fit 20 compounds in one 3D4 well play for 8-point IC50 determination in with and without cofactor and ADPH. From the source play, you can create intermediate concentration by direct dilution from the original compound. Using both source play and intermediate play, you can make your destination play with 8 concentration to run your IC50. And at the very end, you can make human liver microsome dissolved in buffer with and without NADPH and transfer human liver microsome into the destination plate containing compound. This procedure can be saved as a template for other time-dependent inhibition determination later on. So this slide, I wanted to show CYP450 result from the miniaturized time-dependent inhibition assay. Ketoconazole, a known CYP450 inhibitor, show very potent IC50, but no change in IC50 during 30-minute pre-incubation. As expected, ketoconazole show no time-dependent inhibition, whereas verapamil, mibaferdal, and mifepristone show greater than 10-fold shift in IC50 during pre-incubation. These 3 compounds show time-dependent inhibition against CYP3A4. In fact, mibaferdal has been pulled from the market due to mechanism-based inhibition. This slide also shows that data quality is excellent. Every data point is consists of 2 replicates. This slide shows the result for CYP1A2 inhibition assay. As you know, verapamil is a known time-dependent inhibitor against CYP1A2. Verapamil here shows greater than 90-fold shift in IC50 during incubation. So, cost saving for miniaturized time-dependent inhibition. You can expect for running a miniaturized time-dependent inhibition, you can expect to say 100-fold reduction in compound, 5 to 45-fold reduction in human liver microsome. So if you were spending $27 per compound for enzyme, you can expect to spend 60 cents per compound. Of course, there's a saving in substrate, acetylnitrile, and tips. The CYP inhibition assay and time-dependent inhibition assay can be integrated to our automated workstation. ECHO can be used for compound dilution, enzyme and microsome addition, and substrate addition. Centrifuge, bulk dispenser, and reader can be integrated into our fully automated workstation. So in summary, I think we all agree that data quality is the most important factor in assay performance. I hope the data I have shown here have demonstrated that with ECHO liquid handler, miniaturized assay can be accurate, precise, and reliable. The compound potency is also more accurate with ECHO liquid handler with fewer interference from tips, zero dilution, and precipitation. At the same time, you can save up to 90% reagent and also simplify your assay procedures. It is now feasible to run ADME CYP inhibition assay in early discovery stage. So with that, I wanted to thank a few people. These people have worked with me on miniaturization of ADME assays. Sean from Quintera Discovery worked with me on time-dependent inhibition assay. Miguel and Crystal from Merck Serono, we worked on profiling recombinant CYP inhibition assay. Jiong Li from Celsius IVT provide us with a human liver microsome. And Kevin and Tracy from Promega provide us with P450 glow assay, and Joe and Sammy and Brad from Labcyte for their support and suggestions. So next, if there's any questions, I will be happy to answer them.
Thank you, Jing. At this time, we're going to open up for questions. If you've not already done so, please feel free to use the questions window located in the right-hand side of your screen. Please type your question into the window and click the Send Privately button. Jing, we've had a few questions come in as you were speaking. One of them is in the miniaturized assay, some of the, since you're changing volumes, do you also have to change or increase the enzyme or substrate concentration as part of the assay?
No, actually, the concentration of the enzyme and substrate were consistent with the assay in 20 microliter assay volume. So we basically scaled down all the enzyme concentrate, or scaled down the enzyme, scaled down the assay, but keep the concentration of the enzyme and substrate consistent in compared with the large assay volume.
Okay, so there's not an effect of concentration change affecting a difference in IC50, say, like in the example you showed from the Merck data. That's
exactly, yeah. So the enzyme substrate concentrations are all consistent with what they have been using.
Okay. Next question. In the TDI assay, you had DMSO concentration. I think it was lower in the miniaturized format versus the other format. Is it possible to lower it even further, or is that a factor in the assay?
No, you can certainly lower the DMSO concentration. I know DMSO concentration actually affect the outcome, sometimes affect the outcome of your time-dependent inhibition. For sure, because in the miniaturized time-dependent inhibition assay, we had 10 nanoliter compound in the assay. You can lower the DMSO concentration. You can lower the compound addition to as low as 2.5 nanoliter, and certainly will lower your DMSO concentration.
Next question is actually on more of the assay setup. So you switch volumes in lowering from in say a 3D4 format. Do you have to use different plates for that, or can you use sort of the existing plates, or there are special plates that you have to use for that the smaller format?
No, you don't have to have a special plate made for that assay. The plates that we use are commercially available. All you have to do is just make sure the volume that you're using covers the bottom of your well. Make sure your reader is sensitive to read the volumes that you're working with, and that's all. There's no special request for different types of plates.
So that actually is the next question, which is on the reader. So was there any special, there's not any specific, you said commercially available readers was?
Yeah, so we use the BMG reader to read the luminescent-based assay. So there's no special case or there's no special plates, no special readers for these assays.
Okay. And then on reagent actually transferring, so we transfer compound and reagents with a plate inverted. Is there risk of solutions falling, or did solutions, is there anything special that you have to do to prevent that from occurring?
So in my experience, I have never seen any reagent falling off from the plates. For 1536 and 384, the chances for reagent falling off from those plates are very small. But I guess it depends on the plates and then also the reagent. It's a case-by-case. Case-by-case. You want to probably do a little bit of a study before you run a full-blown assay. But so far, I have not seen any reagent falling off from the plates.
So what do you do as part of the study in terms of before you run the assay? So what does that consist of?
So usually, let's say if you have you plan to do, let's say 1 microliter or 2 microliters of reaction. I guess the first thing you wanted to do is you wanted to add 1 microliter or 2 microliters of reagent into the plates of interest or the plates that you're going to be using, and then invert the plate with the reagent inside to see if you have any drops or if you have any drops from the plate. And then if you don't have problem with that, you can for sure that with echo transfer you're not going to have any problem with it.
Okay. A couple other questions are in terms of, I guess one thing that, because the echo uses a single transducer, you're adding reagents one well at a time. Mm-hmm. Do you sort of see that that impacting, say, an incubation time, or how, say, if when you're adding compound into a plate, is that a factor in results?
Right. So that's a good question. When we developed this assay, when we run the small 32 compounds, we took that into consideration too. So it takes longer to transfer enzyme, where it takes much faster time. It takes a lot shorter time to transfer compound. So what you wanted to do is you probably want to transfer enzyme first, then add a compound on top of it. That way you can minimize the time difference when the compound and enzyme seeing each other or react with each other. But so far we have not seen any drift in terms of IC50 change. We've done a full plate evaluation from the first from the first compound to the last compound. We have not seen any IC50 shift from the first compound to the last compound.
Okay, and so again, just to reiterate, you're adding the higher volume reagents first, and then you're coming in later with the compound, which in the one case I think was 7.5 nanoliters, so that's done fairly rapidly after enzymes are already present.
Yeah.
Do you have an issue with mixing in terms of those changes in volume or the difference in volume?
No, I don't have any problem. Actually, when echo add reagent into the plates, there's actually a mixing when you add first reagent and when you come in with the second reagent. There's actually a mixing involved, and especially when destination plate comes out from the echo, there's an invert motion that kind of give you a little bit of a mixing. Of course, we do at the after we add enzyme and the compound, we take the plates offsite and then we mix it for 15 or 30 minutes, depends on your incubation time.
So that actually leads to the next question, which is on incubation time. So you are incubating it in a controlled environment.
Yes. So we actually right after transferring, we take the plates off from the echo, and then we put the plates in the incubator, incubate for 15-30 minutes. However, if you use our automated system, the incubator can be integrated into Access workstation, and it can be incubated on the automated workstation.
One last question. I think this question maybe you've answered this already in terms of the plate types used. So your assay plate was primarily 3A4 or 1536, or your actual assay plate.
Right. So for assay plate, we for both inhibition and time-dependent inhibition, we use a 384. But 1536, we have tested 1536. That works as well. I didn't show the data here.
All of your data today was 384.
384, yeah.
I think that may be the end of our questions for today. With no other questions, I'll conclude the webinar. Those of you who are attending, thank you for your attention and your time this morning. There's a short survey at the end of the session. You'll also receive a follow-up email that will include a link to today's recorded webinar, so you can share that among your colleagues who may not have been able to join us today. Thank you again for your attendance, and have a great day.